for invitation and opportunity to share some of the research projects we do. So I am a professor at the Oregon Health and Science University, and my area of expertise has particularly been treating heritable gene mutations, or at least developing approaches to treat heritable gene mutations in what we call in the germline. Actually, it's before parents pass these genes to children, so we should be able to find somewhere where this mutation is and correct it. Maybe in reproductive cells, we call them germ cells, sperm or egg, or slightly after fertilization. So there are about billion probably people affected with genetic diseases, and most of them actually inherited. That means human population accumulated during so many millions of years these mutations, and now they frequently carry them and then also pass to children. So that number comes from probably contribution from different mutations. We have about 20 or so thousand genes in our nucleus. And each one of them could be mutant. So we know for now for sure about 10,000 or so different mutations that are heritable, and of course because of the it's an error in coding of the protein. Usually these mutations will cause abnormality in the cells and tissues, and will affect depending where that gene is expressed. Most of the time we basically study these diseases and try to link them with the mutation and inheritance, but usually we never can cure them, but ultimate goal of course is to prevent them to transmitting from one generation to another. So we have actually several strategies. Some of them actually actively now used in the clinics. One of them is genetic counseling. So I was trained as a clinical geneticist, and this was actually our main way of communicating with patients and trying to explain what gene or mutation of the gene is causing disease in a child, and how it's inherited, whether from both parents or only one parent. And sometimes we could give them chance, to do something about, whether they're going to have a next child or not. And even if they do, there are some other options besides, just adopting children or not having children at all. So nowadays, they can actually adopt early embryos, which I'll tell a little bit, about this in vitro fertilization. Or sometimes they may opt to adopt the gametes. For example, if the carrier is only one of the parents, so they could adapt to have a different sperm donor or egg donor that would be fertilized in vitro. So more proactive ways is actually prenatal or pre-implantation genetic diagnostics. So it's actually based on, for example, in prenatal, when patients already become pregnant, and during first trimester of the pregnancy, we can actually biopsy a tissue in developing fetus and tell whether it carries that mutation. And of course, if it does, then the option is to terminate early pregnancies. So they actually see based on abortion. Pre-implantation genetic diagnostic is actually a little bit earlier, so these embryos are not implanted yet, but we create several of them, and patients can select the ones that don't have a mutation. But the approach that my laboratory was working is actually to be even more proactive, and rather than just passively selecting non-mutant embryos, actually do something and correct this mutation in carriers embryos. So depending on inheritance, so this is actually shows that how autosomal dominant inheritance works. So sometimes it's just a single copy of the gene. For example, in this case, father carries it, and sometimes a single copy of the mutant gene is sufficient enough to cause disease, despite that there is another copy, for example, of normal copy of that gene. So the examples of this gene would be, for example, some of the genes affecting or causing hypertrophic cardiomyopathy, and we particularly worked with this mutation. Huntington disease mutation is also a single copy is sufficient enough to cause disease. Some BRCA mutations that are causing breast cancer and so on. So in this case, the patient actually will pass this mutation, and just because the mutations are segregated into different sperm, and so but by chance about 50% of children may inherit, where the other 50% may not have it, and this is actually allows us to select early embryos, of course, not children. So how we're going to do early embryos? Luckily, we actually have medical technology that we've been actively practicing, where we recover gametes, egg and sperm from patients, and we could actually fertilize this egg with the sperm in a petri dish in the lab. So these first few days of developing this embryo after fertilization can be actually carried out in incubators in the labs. So this is the so-called in vitro fertilization. Primarily, this technology is used, of course, for infertility treatments. For whatever reason, when a couple cannot conceive naturally, so they will come to in vitro fertilization clinic, and we would recover eggs and sperm, and then do this procedure in vitro, and then we would transplant at about 5 days back into uterus. So this is actually a stage where we could access eggs and sperm and early embryos, and that's where we believe we can actually really affect mutant gene. So the PGD is, or pre-implantation genetic diagnostics, is based on that. Normally, we hyperstimulate the ovary of the carrier woman, and so we retrieve about 5 to 10 eggs, not just one. So that allows us to fertilize them each one separately, and then since we have a window about 5 days while embryo is developing, and it becomes 2-cell, 4-cell. In this case, it's 8-cell. So we can actually biopsy and remove 1 cell in that 8-cell embryo. So the remaining cells are still, would develop normally into fetus. But having that single cell allows us to tell whether this embryo carries mutation or not. So since we have several these embryos, up to 10, so of course we will select the ones that don't have mutation, and we offer them to transfer to patients. Where the other half that carry mutation, normally they become discarded or stored forever in freezers. So this is how a pre-implantation genetic diagnostics works. Even though this is actively practicing medical technology, there are of course ethical issues with destroying already created embryos just because they're mutant. And also, even though we think that we could have a 10, but most of the time we have just 1 or 2 eggs, and of course, if it happens to be the mutant, then we have nothing to transfer. So the idea of gene repair comes from our ability now to induce actually damage in the mutant gene. So we actually did this work recently where we used CRISPR actually to detect and find mutant sequence. In this case, the mutation would be only in one parent. For example, this blue chromosome, it shows that that's a mutant gene. And the CRISPR binds to this mutant sequence, but it would spare the non-mutant, because there is a slight difference in genetic code between mutant, not mutant. And you could design this CRISPR faithfully, only bind and actually make an incision in this chromosome that in the gene where it's a mutant. So that's what usually CRISPR does, and rest of it actually up to cell, what they're going to do with this damage to DNA. And the cells actually, every cell in our body very actively searches for DNA damage, and very quickly they find it because it's actually major damage to cell's integrity. And usually this repair system will start trying to take care of that damage. Most of the time, so we have a kind of gap in the genome, in the stretch of DNA, and the repair system will try to glue it back, so basically make it that one piece again. By doing that, they miss few nucleotides, that pieces of that coding machinery. And then we have kind of induced mutation there. For example, if it was a normal gene, all suddenly now we have a 2 or 3 letters missing. So this type of repair is called non-homologous enjoining, which has been known and actually actively used now in so-called gene editing. But it's good actually to induce mutations. So basically, if you had a normal gene and you want to mutate, for example, in the mice and to see what happens, this is a great tool. And the frequency of this kind of repair has been known to be very high, so cells, for whatever reason, like to repair that way. In this case, up to 99% of repair happens with this kind of error. But of course, we already have a mutant gene, and we don't want to induce another mutation. So our idea was that we want the cells to repair it the right way. So how we do that? So one way to do it is you provide cells with CRISPR, so-called synthetic stretch of DNA. That would be normal. So the hope is that sometimes cells will look for some kind of platform that would be kind of dictionary. That would tell us what's a normal stretch of sequence for this particular gene is, and they will use that actually to rebuild that broken DNA. So this is called the homology-directed repair, and it's been shown in the mouse in other model system before that it happens, but very rarely, so between 1 to 5% cases. So this was actually, it's been known until we started doing it. But we actually, we're hoping that because we have another copy of the non-mutant gene, in this case actually it comes from mother, we were hoping that the embryos would use that sometimes, and we didn't know whether they will use and what the frequency would be of this type of repair. So this is called actually interhomolog repair, which happens to be very active during meiosis, during development of sperm and egg. But this is actually embryo, it's mitosis, and during mitosis, this type of repair thought to be doesn't exist at all. But surprise was that actually after we did this experiment, we found it is actually that interhomolog repair is very high, higher than anything else. So we had actually reported 64% of our embryos repaired using the homolog chromosome, where this non-homologous enjoining was only about, quarter of embryos. But it was surprising that human embryos would never take this external kind of fake gene stretch that we provide. Most of the time, we actually provide multiple copies. There's probably a million of the copies of the small stretches there, but embryos, for whatever reason, or system that actually does repair, recognizes this is actually fake, and they will never use it. This was actually first time we found that even the mouse embryos doesn't seem like differentiate much, but the human embryos, for unknown reasons, actually very picky. So this is actually, we propose the mechanism that can be used to repair genes, particularly when they kind of in heterozygous condition. Heterozygous meaning that there is one mutant copy, but another parent will provide normal copy of the gene. And as I told you, there are 10,000 so genes, and now we're actually working towards testing whether this type of repair can be used to repair all kind of mutations in different genes and types of mutations. The other things to consider, it's actually very high targeting efficiency. By targeting efficiency, I actually mean that whether CRISPR cuts the mutant gene all the time once you deliver it. So in human embryos, it actually could be very high, and especially in some treatments, we had 100% of embryos, for example, who had 100 embryos, and when we inject, all of them would induce cuts. So CRISPR was very effective in finding its target. And again, of course, what happens after a cut occurs is that the repair, we have about, as I said, 64%, and this number actually came with multiple experiments that we did. We actually repeated this experiment on 163 embryos over and over to see if this is kind of one-time occurring event or this is some kind of mechanism. And of course, this is repair, kind of interhomolog repair, it's called, predominantly using the homologous gene present from different parent, from another parent, and this is actually pretty new concept in gene repair, and now it's been actually already replicated. So we just published last year, and it's been already reproduced in MIT lab in the mouse. So we think that this is actually a pretty conserved mechanism. As I mentioned, there are about 50% of embryos we would throw away because they carry mutation. But here we would actually probably salvage at least half of them, and we put them into the pooled embryos that would be transplanted. We actually been doing this kind of germline gene therapy much earlier. About a decade ago, we actually started working with mitochondrial genomes. So as you know, we have two different genomes in our body. One is in nucleus, that's where majority of genes are. But there is a small set of genes actually located in cytoplasm within mitochondria. So this is actually very important genes because they provide proteins that encode complexes that are important to make energy. And so the mutations in these genes also cause, of course, a variety of human diseases. This is actually genes only inherited from mother, so there is no paternal contribution. So one of the difficulties working with mitochondrial genome is actually it doesn't allow to do any repair in it. So we cannot actually edit or insert something. Just that genome is so resistant to any manipulation. So we had actually a very difficult time of trying to come up with a strategy how we repair mutations in mitochondria. And one of the approaches that seems like is working is actually complete replacement. So in this case, if a mother carries this mutation, of course it's on her eggs, so we remove the nucleus, but we'll leave cytoplasm behind with all mutant mitochondria. But of course now that nucleus needs cytoplasm, where would come, and it actually would have to come from another egg. Egg is actually would be donated by healthy woman. So after that, we would transplant that nucleus into this new cytoplasm with donor mitochondria, and then the rest of it would be straightforward in vitro fertilization. So this type of therapy, we call it MRT, or mitochondrial replacement therapy. It's been already tested in animals and actually now actively tested in humans. I think the tabloids call it three-parent baby, if you heard ever. This is a technology that actually refers to this mitochondrial gene therapy. And with that, I think I'll have to thank everybody for listening, and I guess we have some time for questions and answers.
So if you were browsing the New York Times, the BBC, NPR, or just about any other news outlet in the world last August, no doubt you heard about Dr. Mitalipov's work. His team of scientists are the first people in the world to be able to edit a gene in a human embryo, eliminating a disease that could cause serious heart conditions. So it's almost impossible to overstate how revolutionary this work is. And as such, it was even a bit controversial. So were you surprised by all of the press and interest in your work?
I think we actually predicted so this would be a huge interest. First, because it's such a controversial area of research, particularly when it comes to human embryo and mingling with genes, and with all these worries on designer babies. So that's how we know that there will be a huge interest. But besides that, the findings were pretty startling, because this type of repair was thought never exist in mitosis. So maybe during meiosis, when cells become sperm and egg, they become haploid. That's when these type of interactions occur between maternal and paternal chromosomes. But in normal development, for example, in our body, pretty much everybody thinks that these two chromosomes usually actually stay apart from each other and never interact. And here we show that they not just interact, they actually actively repair or present themselves as a repair template, which is quite unexpected. And we're hoping that it's been already confirmed that hopefully we'll see some more research into this.
Mm-hmm. So you have two jobs in a way. You're a scientist, but you're also answering the concerns of people. So what are people's concerns about gene editing technology?
I think the main concern is whether this can be used for something besides just treating disease-causing mutations, meaning for enhancement. Can parents choose to how their baby will be in terms of physical traits or some other things that it's not really disease causing. So it's a kind of enhancement type of gene editing, which is always is of course is a concern, because the technology we're developing probably can be used, because there are so many private in vitro fertilization clinics that practice this medicine, and and most of the time no one controls what they could do.
That's a good point. So the federal government has thrown up some roadblocks just in the case the research wasn't difficult enough. The Congress has barred the FDA from clinical trials involving inheritable genetic engineering, and the National Institute of Health, which is the main funder of scientific research in the U.S. here, is prohibited from funding gene editing in human embryos. These are some pretty major roadblocks, especially in the form of funding what you're doing, which is inherently expensive work. So how were you able to find funding, and what were the challenges?
Correct. So we cannot rely on federal government with NIH. Actually, that ban existed for more than 20 years. I think it's called Dickey's Amendment. That wouldn't allow to use federal funds for any human embryo research, let alone to do, gene editing. And so we actually adapted to do work. Partially some of our research is done by philanthropic support. So as you know, OHSU has a campaign of philanthropic fundraising, particularly for the projects that cannot be, funded by federal government. There are some private foundations, for example, Breast Cancer Foundation, that also provide some funding. So we actually rely on that type of funding. So OHSU is also, of course, it's a clinical enterprise, and there is some clinical revenue. And part of that revenue is actually used to support research that unfortunately cannot be supported externally.
Okay. But other countries in the world do fund and approve of this type of research. So namely Britain, Sweden, amongst others, they've approved using CRISPR technology to edit genes in human embryos. So do you think that this puts U.S. researchers behind the curve of people in these countries?
I think, as long as they don't ban research at all, we still kind of, we can cope and do, and so far we still, I think, on forefront of many of these areas. But I think eventually we probably will lose expertise and people because China or some European countries investing seriously into these type of technologies, and experts probably will choose, particularly my lab members, once they graduate, they just have to find a job in the area of expertise. And since there is no funding, so much restrictions, so they have to just go someplace else. And many of my actually lab members leaving to Asia.
Wow. So federal restrictions are there that are put in place on researchers that are relying on federal dollars. But is there anything to preclude private funding from venture capitalists or from private fertility clinics or other sources from going ahead and doing this type of work on their own?
Yes. So I forgot to mention that the type of research that we're doing is actually probably legal only in Oregon. So other 49 states wouldn't allow it, including New York, where it's, again, I think the most progressive thing, but they still wouldn't allow any gene editing or creating embryos for research purposes. So here in Oregon, about 15, 20 years ago, we had a committee that set the guidelines about what we will allow and what we, of course, under strict oversight. And these type of experiments or research was actually seen as a future development, and so that's why we strive here.
Okay, Oregon leading the way. All right. Let's take a question from the audience. How about on this side of the room?
Hi, considering your comments earlier about other countries embracing the CRISPR technology, are there global regulatory oversights in gene editing research today?
Yes and no. I think there is several guidelines been just set maybe a year or 2 years ago, but actually research goes ahead, and we're actually ahead of any regulatory issues, and so basically they're catching up. But so far, I think the National Academy of Sciences, they actually assembled big panels, and they worked for several months. They actually reviewed some of our mitochondrial stuff before, and now gene editing. And in most cases, they're very supportive, of course, research. Research must go on. But in terms of the clinical applications, they also actually giving us kind of yellow light, they say, meaning if you feel that you've done all the work on safety and efficacy, maybe animals, and if this is the only way to prevent the genetic disease, it must be tested probably under very strict oversight and very small settings.
Thank you.
Okay. So that said, when do you think we'll see the first baby born with CRISPR gene editing techniques applied? Or have they already been
Hard
born?
to predict. Well, I don't think the gene-edited baby was born, but with mitochondrial replaced baby was born. If you probably heard, there was a couple of years ago, even the technology we developed here at OHSU, but we couldn't really transplant these embryos because FDA, federal government, actually regulates, and we're hoping that they will regulate until the Congress actually withdraw funding from FDA to regulate us. So that's how the clinical trials were kind of left there. But because there is so many private IVF clinics operate, even here in the United States, so they actually choose to take some patients to clinics in Mexico, and they did this procedure. Luckily, it was safe, and baby is fine, and so it was genetically cured to correct it. But unfortunately, this is probably not the way it has to be tested. We would like it to be tested in academic settings by experts, not by private IVF clinics.
Sure. We have a question from this side of the room? This side of the room.
Oh, hi. I did my degree in biomedical engineering from Stanford, and this has been an area of interest for me. I guess my two questions are, first, why did you go from the mouse model directly to the humans? Why didn't you go to rabbits or to another organism? And then also, what are your checks for after you've made the cut? And with the example that you were talking about with the mother and putting the homologue into the DNA, and how are you ensuring that there won't be downstream effects? Like, it seems like you've assessed that you've successfully made the cut in the transplant, but how do you assess the downstream effects later if it plays out? I guess is that.
Yeah. So the first question is why we jumped from mouse to humans. Actually, we have a monkey work. So I personally didn't do gene editing in monkeys, but some of my Chinese collaborators already did. And with mitochondrial replacements, all the work has been done in the monkey. As you know, we have a very large primate research center. It's also part of OHSU, which is in Beaverton, and I am faculty there, so I have a research lab there. So we do active pre-testing many of these, particularly gene editing or gene therapy technologies in monkeys before we move to humans. But sometimes it's hard to model, particularly this type of mutations, because they technically don't exist in monkeys. So all these 5,000 monkeys we have in Beaverton, Oregon, they're all healthy. I mean, they probably have sometimes spontaneous mutations, but usually for humane reasons, they usually put down right away. So that's why we cannot have access to, for example, any mutant monkey. So all we have is a healthy. In opposite, in humans, of course, we have so many clinics at OHSU working with a different group of patients, and they have a family history, and for us to find carriers of mutation or patients with mutation was pretty easy. The second question was about unintended, I guess, consequences. Yes, this CRISPR is of course it's a tool that you design it actually on the computer, and you give him this target sequence and tell the computer program to find the best this CRISPR that could bind, recognize, and cut. But because there are some or slightly homologous regions, for this genome, particularly depending, there are some pseudogenes or genes families, and there is always a worry that sometimes CRISPR may cut, some other genes. So it's called off-target effect, and actually one of the reasonable concern on safety. So here, because all these 163 or something embryos, they were not transplanted, so we used them actually to look for not only what happens at the target region, but also if there was any off-target problems. And we've done quite a sequencing the whole genome, and I actually invited top labs looking into these type problems, and so far we actually found none. There was no a single region where we would think that this CRISPR would cut unintentionally.
Well, let me thank Dr. Mitalipov, and I'm excited to see what you'll do next. So.
Thank you.